Let's see here. There's that. There we go. I'll see you guys. There you go. Come on in. It's cold in here. I know it's Groundhog Day. 6 more weeks of winter, says Pete, the groundhog. And then his name, I think it's his name. Seven people, including myself, so 6 students so far attending. And I reckon it's time to get started because it is now 1 o'clock. So welcome you 6, half a dozen students. Today's Tuesday, February 2nd, Groundhog Day, 2021. And 6 more weeks of winter, supposedly. You know, he saw his shadow, so he got scared and ran back into his hole for 6 more weeks. So they say. Well, it feels pretty cold. At least it's cold in my office. But so you guys are biology 1208, section 91, my Tuesday afternoon section. And so without further ado, let's get started. Last time we talked about, well, this is our 3rd meeting, right? Our 4th week, 3rd meeting. Doing our PowerPoint stuff. We did the animal tissues, then the perpetuation of life. Oh, here comes somebody new. And then now we're doing plant tissues. This is this chapter called Kingdom Plant Day. There's all kinds of stuff in it. Huge chapter for a huge, kingdom, Plant Day, all the plants. But in this course, we're really only, at least me, for my course, we're only concerned with flowering plants, because there's other plants that don't make flowers, don't make fruit, they're non-flowering plants. But I just kind of concentrate on these because these are the ones that are most abundant, the ones you're most familiar with. And so we're going to look at the tissues of flowering plants. And they have, let's see, tissue systems, kind of like organs. Animals have organs, plants have tissue systems. And so we're going to look at the tissues in those tissue systems. And so, and give you a little background on flowering plants, okay? And so let me start this. I think I'm going to have to adjust how things are going to look here for a sec. Here comes somebody. I think now we're up to 8 students. There we go. And yeah, this display, I gotta, there, now it looks better. So Kingdom Plantae, the flowering plant tissues. I built this PowerPoint last semester and I'm still using it. And I didn't know if I could use it, all these PowerPoints past last semester, but I guess I am. So but here we are. The flowering plant. What makes a flowering plant? Remember how I said there's tissue systems that make up the plant? Well, flowering plants actually belong to a group of plants that are called vascular plants. And when you say vascular, what do you think about? Like the vascular system, like you have a vascular system, your circulatory system is a vascular system. And what does the vascular system do? Well, it's a transporting system. It moves material from one part of the organism to the next, and that's what your circulatory system does. It moves blood throughout your body. And so certain plants are vascular plants. Now they don't have blood, but they have specialized tissues to help move material within the organism itself, inside the plant. And so those are vascular cells or vascular tissue, okay? And so we're gonna look at that. All your terrestrial plants are vascular plants. That's just one thing that they had to contend with living on land. Originally all life was in the ocean, and then you had aquatic plants, and then once land was available on our planet, the first things to colonize land weren't animals. They didn't have animals, but they were plant-like things. But you had to contend with living outside of water, and so you had to conserve water, and different forms of reproduction and growth and so on and so forth. And so hence the development or the evolution of vascular tissues. Okay, so here I show you somewhere, let me find my laser pointer here. There it is. There's 3 main tissue systems in a vascular plant, okay? There's the ground tissue system, the vascular system, and the dermal tissue system, okay? The ground tissue system is the material inside the plant, the structural cells inside the plant. The vascular tissue system is all these cells that are moving material inside the plant. And of course, the dermal tissue system, like skin, dermis, is what covers and encloses the plant from the outside world. And they all have functions, ground tissues for support, vasculars for transport, and dermals for protection and interfacing with the outside world, okay? So when we'll look at these different parts of the plant to look at the different kinds of tissues that are in there. Now, flowering plants, like I said, they're vascular plants. There's they belong to the kingdom of Plantae, obviously. And then after kingdom, there's a phylum, kingdom, phylum, class, order, family, genus, species, the different taxa of life, classifications, right? Well, kingdom, then phylum. Most biologists say phylum, but botanists, they like to use the word division for some reason. I don't know. A division and phylum is the same thing. Well, I'm not a botanist. I'm not really a plant guy. I like plants. I teach about plants a little bit. I'm by training a zoologist, a vertebrate zoologist specifically. And so I kind of use the zoological nomenclature, terminology, and so forth. So kingdom, phylum, the phylum, you know, in the animal kingdom and the plant kingdom, they're divided into different phyla. Well, there's all kinds of different plant phyla, but all the flowering plants belong to one phylum, okay? Magnolia phyta is the Magnolia phyta, or the angiosperms, which means the seeded plants, the plants that make flowers, or you just commonly call it the flowering plant phyla, okay? And then in that phylum, you know, kingdom, phylum, class, order, family, genus, species, phylum is divided into two classes, okay? And so these are those two classes. So all your flowering plants are going to either be in this class or this class. Those are the monocots and the dicots. And now this table says eudicots and monocots. Just, I don't know why they did that. Just dicots is fine. I have dicots and monocots. But that's what we're going to do. We're going to look at the differences between the monocots and the dicots of the flowering plants in different portions. There's differences you can see in the seeds, the roots, the stems, the leaves, and the flower parts. Okay, and these differences are pretty consistent. Monocot and dicot. That's monocot's short for monocotyledonidae, and dicot is short for dicotyledonidae. What's a dicotyledonidae? All that. Monocot just means one cotyledon. Dicot means two cotyledons. What's a cotyledon? See the one cotyledon, two cotyledons. See that? Cotyledon is basically just the embryonic leaf that develops from the seed. When the seed germinates, in flowering plants, one or two things are going to happen. The seed's going to come up, and it's going to make one little skinny leaf, or it's going to make two little leaves that come up. Monocots make one leaf, dicots make two. Get it? There you go. There's differences in the vascular tissue you see in the roots, and that's what we're going to look at when we look at these tissues. The vascular tissues, they're fall into two types of cells. There's xylem cells and phloem cells. The xylem cells are associated with transporting water, and the phloem cells they transport the nutrients that are manufactured by photosynthesis. And so that's what you got to transport inside the plant, you know, water and nutrients. And so xylem and phloem do that. And how the cells are arranged differ from a monocot and a dicot in the different parts of the plant. In the root, you see this, you get this ring right here. See these rings? The rings are called vascular cylinders. The vascular cylinder of the monocot root forms a ring of xylem and phloem inside the vascular cylinder. However, in a dicot or eudicot, you have a vascular cylinder, but the xylem cells is these large, kind of thin-walled cells here, these red ones. See the red ones are the xylem cells. They don't form a ring, they form some kind of cross, or it kind of looks like a star, doesn't it? It's some kind of way that the cells kind of traverse the center of the vascular cylinder. See that? And they kind of tucked on the kind of like the arms of this cross or star, little patches around it, the blue patches. That's the phloem cells. So this arrangement, this is typical of what you see in a dicot root. Okay. In the stems, you have vascular tissue, but it's not in vascular cylinders. They're in what they call vascular bundles. Bundles, like these little patches. See these little patches of tissue everywhere in the stem. The vascular bundles are scattered kind of throughout, just kind of randomly scattered throughout the stem. The surrounding tissue, that's your ground tissue. We're talking about ground tissue system, a supporting tissue. And this is typical of a monocot stem. And then the dicot stem, they're not randomly scattered. They form a very distinct ring. So this is a vascular bundle. This is a vascular bundle. This is another vascular bundle. The red and the blue, the xylem and phloem. Again, the xylem's the red stuff, the blue stuff. In this illustration, it's just how they're colored. But you look at slides, there'll be different colors and stuff. We'll look at some of that. So anyway, this is the difference in the monocots and dicots in the stem based on the arrangement of the vascular tissues. Okay, and then leaves. They don't show you a microscope slide, but we're going to look at microscope slides of leaves, cut in section, and you're going to see differences. But here, you can see what the differences are attributed to. The vascular tissue in the leaves are the veins. All right, so in the roots, it's a cylinder. In the stems, they're called bundles, and then in the leaves, they're called veins. And so the little streaks right there, those are the veins. That's xylem and phloem in the veins. Okay, and in monocots, in the monocot leaf, the veins all run parallel to one another. See that? But in dicots, they're seldom like that. They're almost always branching. See how they a vein here and a vein here and a vein here, how they branch, and the branches have branches and so forth. So that branching, or what they call net pattern, that's typical of a dicot leaf. Okay, and so these center three examples, the roots, stems, and the leaves, that's concentrating on the vascular tissue itself and the difference. And that's the main part of this lab today is the distinguishing between a monocot and a dicot based on the deposition of the vascular tissue. But there's one other thing, and this is going to be like more next week. We'll look at flowers and fruits and differences in flowers and fruit types. And we'll do the anatomy. Okay. Well, these are pretty consistent differences as well. In a monocot flower versus a dicot flower, and monocot flowers, if you count the flower parts, like the sepals, that's these things in the background, or the petals, these things in the foreground, or even parts of the male and female portion, the stamens and the pistils and all that kind of stuff. But if you count the flower parts, they usually occur in consistent multiples. Okay, for example, if you count petals 1, 2, 3, most monocots have flower parts in threes or multiples of threes, like 3, 6, 9, something like that. However, dicot flowers, you count the flower parts, there's 1, 2, 3, 4, 5 petals right there. And underneath would be sepals, little green ones. And it just so happens that these sepals are the same color as the petals, but it's neither here nor there. But anyway, if you count just the petals, just forget everything else, just count the petals, 1, 2, 3, 4, 5. In dicots, the flower parts, they will occur in multiples of 4 or 5. So it'll be like 4 petals or 8 petals or something like that, or 5 or 10 or 15, something like that. That's typical. Just dicots, it's a characteristic they have. So if you're out there trying to determine you're looking at a monocot plant or a dicot plant, this is the way you can do it. And then there's other consistencies, like the arrangement of the vascular tissues in the different parts of the plant, the cotyledons, etc. So what we're going to do next is we're going to go through, and that's what we're going to do in this lab, is look at the different parts, roots, the stems and leaves, in a monocot versus a dicot, and see the differences. So here we go. This is a section, not magnified very much. This is probably the plants, magnifying in plant stuff is, the cells are huge compared to animals. So right here, both of these are magnified maybe 40 times. And so you can see, here's a monocot root, and this is a dicot root, all right? And the first thing I'm showing you right here in both brackets. Now, I know they're different sizes, but that doesn't really matter. It just so happens to be that this is a monocot root with a vascular cylinder. It's big, but the key thing I want you to zero in on is the arrangement of the vascular tissue in the center. See that ring right there? Those are xylem cells. And then right here This little one right here, it's a vascular cylinder, and that's a little cross in there. That's the xylem cells in there. So the xylem cells form like a ring versus like a cross, or sometimes like it's shaped like a star. Okay. Other parts of the root, inside is the vascular cylinder. Okay. Out here is ground tissue. That ground tissue in this region of the root, from here to here, between the vascular cylinder and the outside, it's known as the cortex. So all of this is cortex, and there's cells in the cortex. Okay. The cells in the cortex, part of the ground tissue, are called parenchyma cells. The word parenchyma means poured in, like a space filler. And like the ground tissue, it's giving support, it's kind of filling that space right there. And in the root, they're also functional in the sense that they'll store the carbohydrate that's manufactured by photosynthesis. All the photosynthesis takes place in the leaves. The leaves essentially are the solar panels of the plant, where you take sunlight and turns it into chemical energy. And so your chemical energy is a sugar, the carbohydrate. It's usually a simple sugar, and like sucrose or something, or fructose, one of those. But it's piped down to the roots by the phloem cells. And then in the roots, it's stored in the parenchyma cells in the form of starch. And starch is a larger polysaccharide that's for stored energy. Like it makes sense. You like in a house that has a basement or a cellar, that doesn't exist in Louisiana, but up north, folks have cellars or basements, and often that's where they would store things underground. Their canned peaches or whatever, or might have a wine cellar. Things that you want to store underground. Your plant, that's where it stores its food, its starch. Okay, this is detail of the Monocot vascular cylinder. So we're zoomed in probably about 100 times here, and so you can see the xylem versus the phloem cells. The xylem cells, they're always, wherever you have vascular tissue, xylem, phloem, the xylem cells are always going to be bigger. Okay, you see that? These are large, thin-walled cells going around like that, forming that ring. Okay, and the phloem cells also forming a ring. They exist. There's actually two different types of cells. We're not going to distinguish between the different types of phloem. So you might talk about that in the lecture. But in the lab, we're just going to go with what we can see. But they're dense. You got like some bigger ones and some denser ones. And they form these little dark. They usually stain a different color, opposed to the xylem. Form these little patches kind of around there. So those are your phloem cells and your xylem cells. Again, the phloem is for the transporting of the nutrients, the carbohydrates, the sugars produced by photosynthesis, and the xylem is the water. It's obtained from the roots, from the soil. Now, it transports, of course, water, but not pure H2O. There's no such thing as pure H2O in nature, because you think about it, the rain comes down, hits the soil, and it trickles down through the soil, and then that water is picked up by the roots and piped into the plant, or whatever. But as soon as rainwater hits the dirt, right? What's in the dirt? There's all kinds of minerals and metals and all kinds of other things in the dirt. So as soon as water hits the earth, there's going to be things dissolving in it. So the xylem cells, they pick up water, obviously, and minerals. So there's things in the soil are going to be utilized by the plant. So xylem cells transport water and minerals. There you go. Again, there's the same thing, but now we're looking at the Dicot vascular cylinder. Remember, that's the one. You can tell it from the Monocot, because remember, the Monocot forms a ring, but the Dicot forms this like cross. Just as long as you have xylem cells crossing the center of the vascular cylinder, you're looking at a Dicot. And that's here, they have larger You know, they got a different color, xylem cells, and then the phloem cells from these little patches, like in the little, on the sides of the arms of the cross. How about that? So xylem and phloem. And then out here and around them, again, would be parenchyma cells again. I didn't label that, but that's what it would be. So there you go. That's the root. All right. Now let's look at the stem. All right. Is a cross section through a stem from a non-woody plant. You know, almost all your woody flowering plants are going to be dicots, but you have herbaceous dicot plants. Herbaceous means herb-like. You know, this doesn't have bark. Doesn't have wood. And most of your herbaceous dicot plants are some kind of wildflower or something like that usually. And your almost all your monocots are some kind of grass. A lot of your grasses and a lot of your iris and lily type things. They're kind of grass-like, you know, that blades, big long blades, like blade of grass, blade from an iris plant or whatever. Those are all monocots, and they're all herbaceous. Almost all your monocots are herbaceous, and a good part of your dicots are herbaceous. But anyway, you look at the section through them, and they're roundish, kind of like a root, but they're different than a root because remember in a root you had vascular tissue, but you had vascular tissue just in one cylinder. But in the stem, you have bundles, and so that's what we're looking at here. These are all these little things everywhere. See little dense little bundles? Those are vascular bundles. That's where the xylem and phloem is. In a monocot, the vascular bundles are just kind of scattered everywhere. And then this is the monocot. And then dicot, the vascular bundles form this kind of ring like this. And so these here. Those are all vascular bundles. Of course, the arrangement is different. So monocot versus dicot. Of course, zooming in to look at the detail, this is magnified about 100 times. Looking at the vascular bundles of a monocot stem, all right? And you can see here's one vascular bundle, here's another vascular bundle, here's another vascular bundle. Don't they look like little faces? Like there's little eyes and a mouth and a little forehead. Sometimes books will call them little monkey faces. You see, or to me, that monkey, they're like aliens. Some kind of little face. If you got a bunch of little faces looking at you, you know you're looking at a monocot stem. The vascular bundles in a monocot stem that makes the little faces. Okay, and the eyes, and I guess the nose, the eyes and the nose, those are all xylem cells. See that, xylem cells? In the forehead, see this little patch right here in the forehead? Those are all the phloem cells. In the mouth, see right there, the mouth right there, the mouth right here. That's not anything. It's just an air space because you got air flowing in there. Okay. So here you go. That's it. This is your vascular bundle, right there with the xylem and phloem. All the tissue around it, again, at the ground tissue, we have vascular tissue, ground tissue, and the dermal tissue. Okay, that's all parenchyma. It's parenchyma cells. Here, it depends on the plant. Sometimes the parenchyma cells have different names, but I just call them parenchyma cells. That's fine. But in the root, the parenchyma cells are mostly for storage starch. In the stem, not so much for storage starch, although they could, but mostly in the stem, it's for support, giving the plant structural support, above the ground, okay, because it's a kind of liaison, a go-between between the roots and the leaves, because we're working our way up to the leaves, right? And that's what the parenchyma cells are. It's kind of connecting the leaves to the roots, and so support and transport. Its function, I guess. Okay, so there you go. Here's a close-up at the vascular bundle in the dicot. Doesn't look like a little face. Okay, this is really close up. And to me, a lot of times when I'm looking at things in biology class, I see, try to make association. What does that look like? To me, the vascular bundles and the monocot stem look like little faces, and the vascular bundles in the dicot stem look like, to me, like pepperoni pizzas. I guess it's kind of a slice of pizza. Kind of, some of them more than others. This is not so much. See, it's kind of pointed, like a cut piece of pizza wedge. Like here's the crust, and then these are the pepperonis. These are all the pepperonis in here. The pepperonis in your pepperoni pizza, those are the xylem cells. Out here forming the crust of the phloem cells. And then everything else that's not part of the vascular bundle will be parenchymal cells. And there you go. That's your xylem and phloem and your vascular bundles of the dicot stem. Okay, there's that. Now, moving on to the root, or I mean the leaves. Again, the leaves, the monocot versus the dicot leaves. This is a good picture showing the veins. See how the veins are all parallel to one another? And that's, that could be a blade of grass. It can be a leaf from a corn plant. Corn, rice, wheat, barley, all that stuff, those are all grains, right? And all your grains are modified grasses. They're all grass. They all have the same kind of leaves with the parallel veins. Your lilies and your irises and things like that have blades. All the veins are parallel, okay? That's all monocots. But everything else have this net pattern, usually have like a kind of an extension of the petiole. The petiole is the name of the stem-like structure that supports the leaf. See that right here, the little petiole. And then see that's like the petiole extends onto the leaf, and then you got branches coming out from it. Well, this extension in the center of the leaf, it's called a midrib. And inside the midribs, there's xylem and phloem. But then you got branches of the xylem and phloem coming off, and of course that forms the veins. So these are the branching veins or the net pattern parallel. Okay, so there you go. And then if you section it under the microscope and look at it, looking at the ends, it's going to look something like this. These little bundles right here, this is magnified about 100 times. Okay, and this is a monocot leaf. This is a dicot leaf, and these are the veins right here. These little bundles, and notice how they're all in the same orientation. That's in cross section because all of them are parallel. Now here it's a little bit harder to see. Here's the midrib, a big extension of the petiole is the midrib. And then the branching net pattern of veins are going to be out here at different angles. And so some of them will be cut in cross section, some of them are cut in longitudinal sections. So it's a little bit different, okay? So this view is pretty easy to see. If you can see this big bulge right here, that's the midrib, and only a dicot leaf would have a midrib like that. And then some of these veins could be larger than others, but they're all cut in the same plane because they're all parallel to one another. But there's other differences we're going to see too, so we're going to zoom in on them. This is magnified probably about 400 times, looking cross-sectional view of a monocot leaf. And then you see right here in the center, that's the vein, all right? And these are the cells on the outside. Sometimes it just called the bundle sheath, because if you think of the vein and a vascular bundle is the same thing, but it's just the outer cells that are supporting the vein, the vascular tissue. And obviously inside, the xylem and phloem. Again, the xylem cells are always going to be the larger, thin-walled cells, those are the ones for water. And then the dense little packed ones, that's the phloem. Okay, so xylem and phloem, vascular tissue, right? There's the vein. And then inside, surrounding, kind of filling the inner part of the leaf, or the ground tissue, it's called mesophyll. Meso means like in the middle. Fill means leaf, like the stuff in the leaf. Well, it's more than just like parenchyma. Okay, the mesophyll. This tissue in the center of the leaf, this is what's responsible for photosynthesis and respiration. Photosynthesis is actually collecting sound sunlight, not sound, sunlight, and turning it into chemical energy. And it requires the addition of carbon dioxide and the CO2. It gets the CO2 from the air. So you got carbon molecules in the air, and the leaf takes those carbon molecules, mixes it with water and sunlight, and turns those carbon molecules into an organic compound, a carbohydrate, and we use that as fuel. So basically the sugar, C6H12O6, is trapped sunlight. It's chemical energy. And that's all that conversion takes place in the mesophyll. Okay. So you got photosynthesis and respiration. Respiration is the transfer of carbon dioxide for oxygen. Oxygen is a byproduct of photosynthesis, and it's released by the leaf. Isn't that nice? Nice for animals, because animals actually use the oxygen to burn for energy. So everybody's kind of got a win-win for everybody, But that's what the mesophyll does. Now here on the outside, and that's part of the dermal tissue system. Remember that? What covers it? The cells themselves are just called epidermal cells. Here they're just labeled epidermis. The cells on the outside, the epidermal cells, enclosing it in. And they usually produce like a waxy layer or a cuticle. You see that? And then also there's modified epidermal cells that open and close across from an airspace. See this right here and here and here? That's an airspace. That's where air comes into the leaf and air can come out of the leaf. The CO2 goes in and the O2 goes out. And regulating that opening are these specialized cells called guard cells. They're guarding the opening, get it? The word stoma refers to the hole itself. The word stoma literally means mouth. And so your leaves have like a little mouth. It opens and it closes. It's more like this. It kind of opens and closes like this, because the guard cells will kind of close together like this, like a gate. And then they'll open, allow the air to go in and out. But if it's a dry, harsh conditions where the leaf might dry out or whatever, those guard cells will close. And so that prevents air from coming into and out to the leaf, because you can lose moisture out of there as well. So sometimes the guard cells help regulate the passage of air and moisture into and out of the leaf. So there they are. And so this arrangement you see right here, with this mesophyll and guard cells and stoma on both surfaces, that's typical of a monocot leaf. Okay? And like you might expect, dicot leaf is different. And there it is. There's different. Dicot leaf, remember, monocot is like a blade of grass. But a dicot leaf, that's like you would see on a tree. Dicots can be woody or they can be herbaceous, but the leaves they're all the same. You're going to have like a tree leaf. You have one side of the leaf that faces the sun, the other side of the leaf doesn't. It's like in the shade. So a sunny side of the leaf and a shady side of the leaf. The sunny side, that's the upper, where the upper epidermis is located, and the opposite of that's the lower epidermis. And one thing that you'll notice is that since this is only sunlight up here, the cells that are underneath it are different than the cells that are underneath or above, I guess you should say, the lower epidermis. So you have two different types of mesophyll. You have palisade mesophyll, that's these guys just underneath. See how they're long and skinny, the palisade mesophyll cells? There's like 2 rows of them right there. These function in photosynthesis only. You look, there's no air spaces up there for gas to move or anything. So these are just for photosynthesis, because that makes sense. They're on the side of the leaf that faces the sun. Okay. Underneath, we got all these little air spaces. That's the spongy mesophyll. Spongy because like a sponge has little pores. Those are little air spaces. Here they call it gas space. Okay. So we're at the gas spaces surrounded by this ground tissue well, the ground tissue in the bottom part of the leaf, that's the spongy mesophyll. So this is for respiration. So spongy mesophyll is respiration, palisade mesophyll is photosynthesis. And of course, notice this, since there's no air spaces up here, you're not going to have any guard cells or stoma on the upper epidermis. It's always on the lower epidermis. So right here, little guard cells and there's a little stoma across from the air space. Always across from the airspace, you're going to have, there's some guard cells right there, no airspace, and so on. There they are. Spongy mesophyll, guard cells, stoma, lower epidermis. And of course, you got your vein. This is a good cross section through a vein. You got your little bundle sheath. That's the outer wall of the vein. And then you can see the xylem cells in there, and little dark green, little dense flowing cells. And this arrangement you see here, common of a dicot leaf. Almost all your dicots will be arranged just like this. Okay, see that? La fin, the end. This is a short lab. Holy cow! I finished in 40 minutes about. It's maybe we should do something else, or maybe not. No, I mean, this is really fast going like this, but in a normal lab, students would be preparing slides and, switching slides and looking at monocots, dicots, the roots, the leaves. It takes a lot longer with hands-on. Showing you pictures is lickety split. So really, that's all we were going to do today. So that was not bad to have a short little lab like that. You guys have any questions about anything? You want to talk about anything from last week or the week before? We can do. We got all kinds of time before I let you go, or we can just go. No questions, none of you. So what are we going to do next week? Next week, let me show you this. Let me get this out the way. So here's today, flowering plant tissues. I do a little, again, all my labs, I kind of turned them into PowerPoint pretty much. And next week, we're going to do same in this chapter, but we're going to look at the flowering plant anatomy. And so we're going to look at different types of roots and stems and leaves, et cetera, of, like the gross anatomical view. And look at that. I think I have a PowerPoint presented for that. We'll go over that. And then the week after that's Mardi Gras. And then the week after that's flowers and fruit. And the week after that is your practical. So we only have like 2 more meetings before your practical. So your practical is going to be over this PowerPoint, animal tissues, then flowering plant tissues, plant anatomy, flowers and fruit. Oh, and perpetuation of life. How about that? Yeah, those 1, 2, 3, 4, 5 things. And that'll be it. And I think I already scheduled your midterm on my courses. It's up there, whatever the date is. I can't remember. But we still got a few weeks. But only basically 2 labs because we're squeezing in Mardi Gras. So, but any questions? You good?
Just one. I just missed one thing. In the beginning, when you were talking about the 3 main tissue systems in a vascular plant, and you said the dermal tissue system is protection and interfacing with the outside world, I missed what the other 2 were when you quickly summarized that.
Well,
so you said ground tissue system and the vascular tissue system, and you just gave like small summaries.
Well, the vascular tissue system, that's where the xylem and phloem. That's for transporting water and nutrients inside the plant. So that, that's its function. And then the ground tissue is, the space, the stuff that's in there. Basically everything other than the xylem and phloem in the roots, the stems and leaves. In the roots, like the parenchyma cells, would be part of the ground tissue. And in the root, it'd be storage. In the stem, it's space filler, but actually support of the stem itself, and kind of acts as a go-between of the leaves and the roots, supporting the vascular tissue, etc. And then of course in the leaves, it's part of the mesophyll. The mesophyll, filling the interior. But the mesophyll, whether it's just regular mesophyll or spongy mesophyll or palisade mesophyll, it's associated with respiration and photosynthesis. And then the dermal tissue, that's the stuff on the outside, like the epidermal cells. And the guard cells will be part of that as well. So, does that make sense?
Yes, sir. Thank you very much.
Okay. Anybody else? Is it all good? All right. A lot of that, I'm glad you asked, because it's kind of fun to talk about. But most of that kind of information, I know I talk about that, and I'll go into more detail, just explaining things. But to let you know, like in my lab's all identification. I won't ask you. Those are more, in my opinion, those are more like lecture type questions and content. My lab is, I'm going to show you pictures and say, what is this? What is this? What is this? What is this? I'll point to a cell, say, name this cell. Is it a xylem, is it phloem, is it parenchyma, whatever. Or name this, is this a monocot root or a dicot root? Or is it a monocot stem or dicot stem? Kind of like that. So it's all recognition in the lab. So in my lab.
Yes, sir. I just asked because I was curious.
Okay, no, that's fine. Curiosity is good. Anybody else? Okay, guys. Well, I will see you next week, and we will talk about flowers and fruit. Is that right? No, I'm sorry. Flowering plant anatomy. That's next week. All right. See y'all then.